Hello everyone, my name is Dennis and today I'll be talking about QSAR. What is QSAR? QSAR stands for Quantitative Structure-Activity Relationship. Before you stop listening because this sounds too complicated, let me explain in easier terms. In chemistry and biology classes in high school, we learned that structure determines function and properties. Whether it's a structure of a simple molecule, structure of a protein, or structure of an entire organ, if looking at a large enough number of objects with that specific structure, we can start determining functions of the thing that contains it, that's QSAR. Noticing the presence of carbon with 3 hydrogens around it makes a molecule likely to be more undissoluble in water is QSAR. Noticing the larger molecules weight more is also QSAR. Now you might ask, why would we spend time to find a relationship between structure and property if we can just directly measure the property? Most of the time, this allows saving time and resources. Let's say you're an owner of a good company that's trying to develop a plastic that is biodegradable and photodegradable to wrap your goods in. You want something that will degrade within a year in a bacteria-rich soil on the presence of sunlight to prevent the environment from the harm of plastic pollution. Usually we'd have 100 candidate materials that you then test for strength, elasticity, water permeability, photodegradability, biodegradability, and so on. You would need to test each one of these parameters in a lab on a large enough set of samples to believe that your results are consistent. Now let's do some quick math. 100 materials times 10 samples times 5 possible properties to be tested gives you 5,000 experimental runs that you have to perform. Now let's bring QSAR into this problem. Let's say that you developed a biodegradability model that with 90% accuracy can predict whether the material is biodegradable or not. You also develop a similar photodegradability model. You then use your models on 100 plastics you're trying to test and see that only 50 of them are biodegradable and 20 are photodegradable, with 15 of them overlapping between two. The math becomes as follows. You test 15 materials times 10 samples times 5 possible properties. Just a quick sort with our modeling brought the number of necessary experimental runs from 5,000 to 750, greatly reducing the cost of our product development. Because the model is never perfect, we still need to check the actual bio and photodegradability. As in theory, some non-degradable materials would sleep through each model. Now that you understand how useful QSAR models might be, let's dive deeper into the process of their development. Sometimes the patterns are obvious and could be found right away, like saying that round objects are easier to roll than triangular. However, many patterns are not obvious, even to the most trained experts in a field, and require extensive analysis of large datasets. Let's talk about what building a linear QSAR model of biodegradability would look like. We start by gathering experimental biodegradability data of a large enough number of molecules. We then come up with a variety of ways to describe those molecules. Their weight, shape, electrical charges at various regions, presence of some smaller well-known molecules within them, and so on. These are referred to as descriptors. They're usually pre-made and readily available on the internet. Once we gather necessary data, we start the modeling process. We then split all molecules into two groups. 80% usually go into the first group, called the training set, and 20% go into the second group, called the test set. The reasons behind doing this are explained later. Once the data is split, we plot a descriptor versus biodegradability graph for the training set. We then draw a line of best fit and measure how well the line fits all the data points, a value also known as R-square value. If R-square is high enough, preferably higher than 0.65, we keep the descriptor. We then plot the test set data points and draw the line of best fit from the previous step. In this way, we ensure that the line works well with any type of molecules and not just molecules in our data. This is called overfitting, when model is too focused on the data it's trained on. Model would be extremely precise on a specific data, but might be useless for other molecules. By splitting it, we make the test portion imitate the unknown molecules, since the line of best fit is not trained on it. Then we repeat the same procedure with every other descriptor. After gathering all the good ones, where R-square is higher than 0.65 and R-square test is within 0.07 of 0.65, we combine them into the complete model. For example, biodegradability could be modeled with 72 to 76% accuracy using the simple following formula. There are other types of modeling other than linear, most of which have a higher accuracy. However, linear models are easily interpreted and shed light on questions of science. For example, from the formula above, the presence of alcohol in the molecule makes it way more biodegradable, the property that might not have been easily found without aid of machinery. Thanks for listening and keep learning.